Now it's my great honor to introduce our next speaker, Dr. Jennifer Mitchell. Dr. Mitchell received her PhD from the University of Toronto, conducting her research on transcription factors regulating gene expression in the uterus during pregnancy at the Lilienfeld Tannenbaum Research Institute with Dr. Stephen Lai. Dr. Mitchell then pursued postdoctoral training at the Babraham Institute in Cambridge in the UK, where she worked with Peter Fraser to investigate how genome organization regulates transcription in erythroid cells. Then in 2009, Dr. Mitchell started her own research group at the University of Toronto, where she investigates transcriptional mechanisms regulating pluripotency and lineage commitment during development. As an independent investigator, Dr. Mitchell has produced some exquisite work dissecting the mechanisms by which transcriptional regulatory elements activate gene expression in stem cells. And I'm very excited to hear the latest on her research in her seminar today. So please join me as you can and give me a warm, if virtual, welcome to Dr. Jennifer Mitchell.
Great, thank you so much for that introduction and for the invitation to speak today. I'm going to mostly tell you about some of our most recent work that was actually just published in Genome Research last week. And in this work, we were really trying to understand what are the sequences that actually drive the activity of enhancer sequences in our genome. Okay. So as I'm sure you're mostly aware, the human genome is vast. It's huge. So around 6 billion letters. And in fact, most of that is non-coding. And we really only understand the meaning of the sequence for about 2% of the genome. So by that, I'm referring to the coding regions. And what I'm showing you in this image at the bottom is what the print version of Wikipedia would look like, which is around 3 billion words. So just to kind of give you an idea for comparison to the size of our genome. And then here I'm showing you what about 2% of this looks like. So we can really only understand what the sequence means in this very small portion of our genome in those coding regions. And of course, we don't know everything there, but we at least understand how the bases translate into function. But for the non-coding regions, we really don't understand the sequence at that level. So in the coding regions of the genome, like I said, we understand how individual bases are transcribed and then translated into specific amino acids in proteins. These regions of our genome are usually highly conserved. We know that every base is important in creating this information, and we understand that code. And that gives us sort of a starting point to then further explore and understand function. But in non-coding regions of the genome, and specifically, I'm going to focus on sequences that regulate gene transcription. Usually these have fairly low sequence conservation across different species. We know that most of the sequence in these even important regions is actually not functional. And we can't really identify very easily the functional units within these regulatory regions, even when we know where they are in the genome. So this is a really big gap in our knowledge that limits our understanding of how our genome works, how the sequence of our genome leads to specific functional properties and can sometimes be associated with disease. Of course, these transcriptional regulatory elements are really important for functional properties of our genome. We know that they are generally associated with open chromatin. So I'm representing that here by the absence of nucleosomes in this enhancer region. We also know that they are bound by transcription factors that recognize specific sequences within this open chromatin. Those transcription factors recruit coactivators, some of which modify histones, adding specific histone modifications that are often located within enhancer regions. For example, histone H3, lysine 27 acetylation, or histone histone H3 lysine 4 monomethylation are most often associated with enhancers. And they also recruit other important regulatory complexes like the mediator complex. And together, this facilitates the recruitment and activation of RNA polymerase to the gene promoter. We know these enhancers are really important for tissue-specific expression. And what I'm showing you here is an example of a reporter assay that was included in the VISTA enhancer browser, showing a specific sequence in the genome that drives expression in the developing forebrain most strongly, and also in other neural tissues to some extent. We know that these enhancer regions that can be located at very long distances away from the genes that they regulate tend to form these architectural interactions with gene promoters that they regulate, sometimes referred to as chromatin loops, and that this is important for regulating specific target genes. And then we also know that deletion, duplication, or mutation within enhancers can cause disease and also alter phenotypes. So here I'm just giving you an example of the beta-globin locus. And the reason why I'm talking about this is that this is really a distal regulatory region that has been studied for more than 50 years. And although we know a lot about how gene regulation works for the beta-globin locus, we still don't have a really complete understanding of how these genes are regulated. What I'm showing you here are the beta-globin genes, which this is a cluster of different beta-globin encoding genes that are regulated in a developmentally controlled manner. And we know that upstream of these genes is what's been called the beta-globin locus control region. And this was first identified because deletions of this region cause a rare blood disorder, beta thalassemia, due to low expression of all of the beta-globin genes. And then the other thing that I'm showing you here are some genome-wide association study identified single nucleotide polymorphisms, or SNPs, that are found within the LCR and that are linked to hemoglobin concentration in the blood. So although we know a great deal about the regulation of the beta-globin genes, we still don't really understand how specific individual SNPs within this enhancer are driving altered function of this region. Of course, there are many, many other examples where genome-wide association study linked single nucleotide polymorphisms are linked to disease. And in fact, the GWAS catalog contains close to 200,000 unique SNP trait associations from close to 5,000 publications. And in fact, the vast majority of these, more than 90%, are not in gene coding regions. And many of these non-coding SNPs are predicted to affect gene regulatory elements. But at the same time, there are only really a few select GWAS SNPs that we actually understand at a mechanistic level. And so by that, I mean, do we know what the target gene is? Do we know how the SNP alters function? Sometimes we don't even necessarily know in what tissue context this misregulation occurs. But we have this sort of common hypothesis that's been put forward for how these non-coding single nucleotide polymorphisms are leading to disease, in that in some cases, these regions might be bound by specific transcription factors, causing an architectural association with a target gene, and that this is the sort of normal healthy situation. But in the disease context where the SNP is present, then perhaps one of those transcription factors fails to be recruited, and that might disrupt the architectural association with the gene itself, or just affect the expression of the gene, causing a disease situation. Of course, the reverse is also true. where the healthy situation might be a lack of an association of a specific activator. And in the disease situation, the gene might be aberrantly overexpressed because of aberrant recruitment of additional transcription factors. And I'm going to propose now that a better understanding of how the sequence code within gene regulatory regions is actually leading to function would support a better understanding of these mechanisms that are linked to disease. And so first, I just want to give at least my sort of opinion on one of the reasons why we don't have a better understanding of what the regulatory code is. There are lots of reasons for this. This is a very complex problem. And I think we're still going to be working at it for quite a while. But one of the reasons that I think we're lacking some information is that a lot of computational investigation into sequence features has focused on regions that are bound by one transcription factor. And so I'm showing you an example of that here. So here's the cell1 gene. And then downstream of the gene, there are locations where the SOX2 transcription factor binds to the genome. This is based on ChIP-seq data for that protein. So it's actually identifying bona fide binding of this transcription factor. And if we just look computationally at the sequences that are found at these regions across the entire genome, what we get out in the case of pluripotent cells, which this is the example that I'm providing you, is we identify the sequence bound by SOX2, but in this case, we also find this OCT4 SOX2 co-motif that tends to be bound in pluripotent stem cells because SOX2 binds with OCT4. And that's really important and useful information. But if we would now go back and scan this small region of the genome for this type of a motif, we would find that motif all over the place. So it doesn't actually really tell us what about these specific regions is causing the binding of this factor. On top of that, there's another layer of complexity here, because if we actually now take these regions bound by SOX2 and we investigate those regions for whether or not they actually have enhancer activity using a reporter assay, what you will see is that some of the regions are enhancers. So I'm showing you here that 2 of these regions do function as enhancers, but actually many of the other regions are not actually enhancers. So they don't have the ability to drive transcription of an associated reporter gene. And now you can sort of see that if we're looking at all of these regions as a group, we're sort of clouding our computational analysis by having false positives in our group of regions that we're looking at to try to understand the regulatory sequence code at enhancer regions. So we also noticed when we looked a little bit more carefully at other data that was available for these pluripotent embryonic stem cells, that if we looked at the binding of additional transcription factors, then those 2 regions that actually are enhancers quite nicely lined up with the regions that were bound by the highest number of different transcription factors. So suggesting to us then that a larger number of transcription factors are required for enhancer activity. But we sort of ask the question, well, how many are actually required? And part of the motivation for this work came out of this particular region here, which you can see is actually still bound by a number of transcription factors, but is bound by a couple fewer than those active regions. And despite the fact that this region binds several master regulators of pluripotent transcription, this region does not have any enhancer activity. And so this sort of question, why is that the case, is part of what motivated the work that I'm sharing with you today. So to try to answer this question, we focused our investigation on non-coding regions that were bound by multiple transcription factors. So instead of just looking at the regions bound by one factor, we focused on regions bound by multiple factors which also had chromatin features associated with enhancer activity. In addition to that, we took advantage of the fact that for pluripotent embryonic stem cells, we have data from both mouse and human that describe where transcription factors are bound throughout the genome. And so we decided to focus in on regions like this one that I'm highlighting right here, where in both mouse and in human, a number of transcription factors are bound to this region, because we hypothesized that it would be easier to identify the important sequence features driving activity if we looked at these regions showing evidence of conserved function in both mouse and human. Okay, so we took a clustering approach. We identified all of the regions bound by at least one transcription factor in mouse embryonic stem cells. And then we looked at how transcription factors were associated with the genome in both mouse and human. And we also looked at the H3 K27 acetyl signal for both mouse and human. This was close to 25,000 regions, and we clustered these regions using these 20 features. And what we noticed immediately is that there was a fairly small proportion of these regions, close to 10%, that were bound by multiple transcription factors in both mouse and human pluripotent stem cells. We called these CHEF regions, for conserved high enhancer feature regions, and these were the ones that we mostly focused on for our sequence analysis. These regions alone do not describe what we think are mainly the active enhancers in mouse embryonic stem cells, because of course there is also a set of what looks to be mouse-specific enhancer regions, high enhancer feature regions shown here. And together, these two groups represent about 20% of the transcription factor bound regions for this cell type. And based on our evidence, as well as reporter assays that are published from many other groups and some recent STAR-seq genome-wide approaches, most of these regions that we call low enhancer, conserved low enhancer feature regions, or CLEF regions, we think that most of these regions are actually non-functional. So these are the regions that bind only one transcription factor, usually in only one species, and are also associated with fairly low levels of H3 K27 acetylation in both species. I'll also make the point that all of these regions contain open chromatin. So these would all be identified as open regions by, for example, an ATAC-seq assay. So again, we think that quite a small proportion of these regions are actually bona fide active enhancers based on our work, as well as the work of many other groups. Okay, so focusing in on these CHEF regions, or the conserved high enhancer feature regions, we first just wanted to look at overall sequence conservation. And as we expected, we did find that these regions are significantly more conserved between mouse and human than are the other regions that were identified as clusters. And also they're significantly higher sequence conservation than random regions. So this was a good indication that in here were important sequence features that we could identify using computational approaches. What are these conserved sequence features? Well, as you might imagine, these are largely transcription factor binding sequence motifs. I'm showing you an example here of an OCT4 SOX2 site that's highly conserved between mouse and human within one of these regions. And in fact, if we look at all of the transcription factor binding sequence motifs in the JASPAR database, we find that not only are most of the TFBS associated with proteins for which we use ChIP-seq in our analysis are conserved, but also shown in blue here, many other TFBS are also highly conserved in this set of regions. And these represent TFBS for which we did not have any ChIP-seq data available at the time. So again, this is telling us that there are additional sequence features that are in these regions that we actually don't yet have information on for this cell type. Also, on average, if we can compare those conserved high enhancer feature regions to the regions bound by only a few transcription factors, we can see that these regions do, on average, have a significantly higher number of transcription factor binding sequences, but it's not a huge difference from what we think are those non-functional regions. So they tend to have around 12 transcription factor binding sequences on average, whereas what we think are the non-functional regions have fewer than 10 of those transcription factor binding sequences. So again, this is telling us that there is a difference between these two, but it's not sort of a night and day. Yes, we contain transcription factor binding sequences, and no, there's nothing that we could find in here. And now I think this finding was, again, what sort of highlighted for us how complex this problem is, to try to understand why these regions are binding a large number of transcription factors, whereas these ones are really only binding one. Okay, so to now kind of continue with our computational analysis, we decided to expand our approach of using multiple genomes in our analysis. So initially we had only been comparing between mouse and human, and now we decided to compare between 6 different species. And we used this previously published tool called Motivo to identify both the transcription factor binding sequences that were present in multiple species and the ones that were highly conserved across those multiple species. We also used a lasso regularized logistic regression machine learning approach to identify which specific transcription factor binding sites would distinguish those high enhancer feature regions from other clusters. And what we found from that approach was we identified a much more complex set of transcription factor binding sequences than we were originally expecting. And what you can see here is a heat map of how well enriched specific TFBS are in those chef sequences. And we identified a group of more than 70 different TFBS that are enriched in chef sequences compared to the low transcription factor bound regions in our set. We did also interestingly identify a few specific TFBS that are actually depleted from these regions. And we think that some of these represent negative regulators of enhancer activity. But we focused most of our next approaches on what we think are the active regulators of enhancer activity in this cell type. So some of these TFBS are the ones that we would expect based on the chip data that we originally used, but we also identified several novel TFBS. And for some of those novel TFBS, there's recent evidence that they are important in embryonic stem cells. So this was, again, comforting to us that our computational predictions were lining up with some new evidence that was coming out in the recent literature. I just want to take you back to that region that I initially said was part of the motivation for this study. Remember, I showed you that downstream of this SAL1 gene, there were individual regions bound by SOX2, but only a few of these were active enhancers. And then I highlighted this one region down here that is bound by a larger number of transcription factors, but yet still lacks enhancer activity. And so something that we did to try to see whether or not we were on the right track with our thinking about this is that we took this inactive region that is bound by many transcription factors, and we added in a few different transcription factors from our computational analysis to see if we could engineer activity. And so the first set we chose to add in was 3 factors, ESRB, TFCP2L1, and SMAD3. And you can see now that we can turn this inactive region into a really strong enhancer. by the addition of 3 different TFBS. Now, if we only add 2 of these into this region, actually, we don't really gain a lot of activity. And this result started us thinking that, well, there might be this sort of threshold number of TFBS that you need to have within your region to take you from an inactive region to an active region. And another thing that we did is we added a different combination. So we still added ESRB and TFCP2L1, but we added this other motif for E2F1. And again, we saw increased activity in this case. And so this indicated to us that there is also likely to be some flexibility in which of the TFBS that you can add to this region to cause activity. So just by way of summary, we had found that these high enhancer feature regions contain on average 12 different conserved TFBS. And we found this larger repertoire of 70 different TFBS that are predicted to bind to different TFs. And so we decided to now take that information that we had discovered and see if we could actually use those features to build a synthetic enhancer, to say, do we now have enough understanding of this regulatory code to just try to build something from these principles? So what we're doing is we're taking the TFBS that were enriched, combining them together with just 2 base pair short spacers. We're doing random TFBS selection and random ordering. So we're not really worrying about the order or orientation of these sequences in our synthetic constructs. And of course, it's really important to benchmark these approaches against other information that we already have. So for example, we wanted to compare our synthetic sequences to the strength of a native promoter and a native enhancer. And what I'm showing you here is the enhancer activity of the enhancer that drives SOX2 expression in these cells. We also wanted to benchmark our synthetic sequences against some more recent massively parallel reporter assays. So this one is from Fiore and Cohen in 2016. And what they did is they took 4 transcription factor binding sequences that we know are important in this cell type, and they combined them together in different orientations and orders, and they assayed more than 600 sequences for their activity. And so we chose actually what is the highest activity region from their assay, and we compared it against our native enhancer. And we actually found that this region has very limited activity when you compare it to a native enhancer, despite the fact that it contains 4 transcription factor binding sequences for different transcription factors. What I will say that what was really important that was shown from this work is that having different TFBS included in your construct is actually really important. So if you just repeat the same TFBS multiple times, which is what's shown here, you don't tend to see much activity, but where you see more activity is where you have all 4 different TFBS. And we use that information in our design approaches as well. This is also more representative of what we see in natural enhancers, where they don't tend to contain repeated TFBS very often, but they tend to have a variety of different ones. So to also sort of investigate whether or not the top TFBS in our computational prediction was just maybe the most important thing that we found, which in fact happened to be that OCT4 SOX2 comotif, we actually took that one and repeated it 14 times. And again, we found very limited activity for this repeated TFBS motif, despite the fact that this sequence should very strongly bind the 3 master regulators of pluripotency, OCT4, SOX2, as well as NANOG. So now the regions that we designed based on our computational analysis, we took any of these sequences that were enriched in our chef regions and we combined them together randomly and with random ordering. And we were able to create 3 strong synthetic enhancers that drove a similar level of expression as did our native enhancer. And then we also took 14 TFBS from these either depleted or not enriched set of TFBS. And although these are all for transcription factors that are actually expressed in embryonic stem cells, we do not find that there is any activity associated with that region. And so this tells us that just taking any TFBS for expressed transcription factors is not enough, that you have to actually learn what this repertoire is. And so based on this, we think our understanding now of this sequence code is that there's a large repertoire of pluripotency associated transcription factor binding sequences that bind specific transcription factors. And also we learned that specific master regulators are not absolutely required, because this first construct here included the OCT4 SOX2 co-motif, whereas these other two constructs did not. And so we know now that that co-motif is dispensable for activity, and those two constructs also did not contain any OCT4 or SOX2 motifs. Okay. So I alluded to this earlier, but because we found that the low enhancer feature regions contain just slightly fewer than 10 TFBS on average, whereas the high enhancer feature regions contain more than 12, we were thinking that there might be this sort of threshold of TFBS that are required. And so to look at that more directly in an experimental approach, we chose one of our strong enhancers and we started to erode TFBS motifs away. And what we found was that if you take a few away, not very much changes. But now if we take a few more away down to around 10, we see very limited activity. And if we take a few more away down to 7 TFBS, we see absolutely no activity, despite the fact, again, that this region contains 7 different transcription factor binding sequences for a few of the important regulators that are known to be involved in this cell type. We also looked at this concept of a threshold using a native enhancer. And this is the enhancer region that drives SOX2 expression, that transcription that I mentioned earlier. And what you can see here is all of the computational predictions we made for these TFBS that were conserved between mouse and human. And we were able to mutate a number of these, and all of them are not functional, but actually most of them did. Mutation of most of those TFBS did affect enhancer activity. And shown here at the very end, when we mutate 2 of these, we lose almost all enhancer activity. Again, telling us that you really need a large number of these TFBS within one of these enhancer regions. Okay, so why are so many of these TFBS actually required? Well, we think that what is actually happening here is that individual transcription factors are able to perform able to perform different roles within these enhancers, and that you need enough of these roles being carried out within a sequence to actually confer function. And what I'm showing you here is an example of different transcription factors that can recruit different coactivators, that can directly associate with polymerase complex, or that can directly associate with mediator complex. And I hope you can appreciate that it's not every factor within this set that can do all of those things, but that there is some subspecialization of different transcription factors being able to, for example, very strongly recruit one of the components of the mediator complex. And we think that if you need to assemble this really large mediator complex of around 30 members, then it's important to be recruiting multiple different parts of that complex to really appropriately assemble this large multi-protein complex. Okay. What we're doing now is we're working on making these types of predictions for other mature cell types. We have done this approach in cardiomyocytes, in neurons, and in liver cells. And I'm hoping that you'll see that work being at least posted soon on bioRxiv, but we're not quite ready to post it yet. So I just thought I would let you know that that's what we're working on now. And in this case, because these are differentiated adult cells, we're able to more directly look at those disease links that I talked about at the beginning. And so just by way of summary, we're hoping that this improved understanding of the regulatory code will allow us to better understand those disease links in the genome. We've shown that analyzing regions with conserved transcription factor binding does identify the important transcription factor binding sequences and thereby regulatory transcription factors that were previously unknown. It also allowed the first construction of synthetic enhancers with activity similar to native enhancers. And we hope that we'll be able to use this to inform a better understanding of disease links. And so I'm just showing you here this image of a physician sort of saying to a patient, oh, look, here's your problem. We're really not here yet, except for with a very few mutations in coding regions that we really understand at a much deeper level. For most of the differences in our genome that are linked to disease, we really, don't have this level of understanding yet. And then just as a way of acknowledgement, the people who did most of the work in my lab, most of the lab did contribute to this work, but mainly it was Gurdeep Singh who did all of the computational analysis that I presented, and also did a lot of the wet lab experiments as well. He will be defending his PhD thesis in a few weeks. And also Chanel Moulani did a lot of the work with Gurdeep when she was an undergraduate student in my lab and has now joined my lab for a PhD. And so she will be working to continue on with our understanding of these regions. And then this project is almost exclusively supported by NSERC funding for my research lab. And thank you everybody for your attention. And thank you to the rest of the members of my lab that contributed to the work. And I'm just going to end with my slide here, which was a artwork that we commissioned based on our findings. And so I thought it would be nice to leave this up at the end.
Awesome. Thank you, Jennifer. That was really cool. They've got a whole ton of questions, but I'll start with Marco has raised his hand. So I'm going to unmute you and you can ask your question.
Can you hear me now?
Yes.
Nice. Hi, Jen. Beautiful, beautiful stuff. Two questions. One is, if you, in these regions, they are bound by few transcription factors and do not have any reported activity in your assays, do you have evidence if they have enhancer RNA transcription from them? And the second question is, what are these regions, right? These are still bound by transcription factors, less than 10. Apparently, they are not enhancers. So what would you speculate they are? Thank you.
Yeah, so was your first question, do they have enhancer RNA?
Yeah, exactly.
Yeah, so the ones that are really only bound by one transcription factor mostly do not have enhancer RNA. But we have identified regions that have no enhancer activity at all, even by CRISPR deletion. So I didn't show you that just for sort of simplicity in this shorter talk, but we have also done CRISPR deletions on some of these multi-transcription factor bound regions that seem to be non-functional in reporter assays. And also by CRISPR deletion, they do not alter transcription of the only gene that changes expression when we delete a larger region containing all the enhancers. So, and that cell, one example that I gave you, that does have enhancer RNA, but yet it seems not to be functional in this context, at least. So then the other question is, well, what are they doing? So I think there's lots of ways to answer that. You know, one way to answer it is just that, well, binding does not necessarily equate to function, right? So just because one transcription factor binds doesn't necessarily mean that region has to, quote, be doing anything in any way, right? I think it's perfectly logical to think that there might be binding of transcription factors in our genomes, and those regions may have no function whatsoever in that context. And even the binding might not be important in any context. It's just that because of the sequence features that are there, that factor is able to bind, but it doesn't cause any problems because it's not causing any spurious activation.
Why would the chromatin be accessible if that's a non-functional binding?
Well, you know, why indeed? These proteins have the ability to associate with the DNA, depending on the sequence. SOX2 itself is a pioneer factor that can cause opening of the chromatin. So, okay, so that's one answer. And I think that in some cases, that is probably the case. Probably there's some binding that's totally non-functional. In other cases, it could be that, you know, these regions really are poised for some opening later on. Maybe there's, you know, this factor is kind of sitting there waiting to be joined by some other transcription factors when the situation changes, right? And I think that probably both of these things are going on. I don't believe that every single incidence of binding in the genome has to be functional, but I do think that there are definitely going to be examples where binding without function in a particular context is important for function when something else happens, whether that's differentiation or whether that's, you know, signaling mechanism that activates something new that the cell then responds to. So I think both of those things are probably going on.
Thank you, and congrats. It's a really beautiful study.
Thank you. Thanks for your question.
Okay, so we're after one, so I'm going to ask one last question, but then we have the coffee chat after that. So for everyone else who didn't get your question asked, because there's many of you, please just post in the chat that you want to stay for that, and we'll promote you to panelists, and there can be a continued discussion there. But this question is from Irene Kaplow, and she says, "Thank you for a fabulous talk. For the regions bound by many transcription factors, do you think that all of the transcription factors are bound at the same time? Or do you think that only a few are bound at a time, and the presence of many binding sites ensures that something is always bound in spite of many of these factors having low expression?"
Yeah, I think that's a really interesting question, and it kind of takes us, you know, over to biochemistry, which is not my area of forte in terms of the lab, the type of experiments my research lab does. But how I envision this is that, yes, there is a lot of on and off. And so I don't think these things all have to be bound at the same time. I see this a little bit more in terms of how, you know, we started thinking about things in Peter Fraser's lab where we were thinking about more transcription compartments and also kind of loose organization. So I think of this as the DNA as kind of being this recruitment platform. And these transcription factors, you know, might be coming on and off, but that creates this sort of local high concentration of many factors. And that local high concentration of those many factors then recruits in other functionalities, like those coactivators I mentioned, like multiple components of the mediator complex, and also facilitates recruitment of the polymerase and other components that are needed to actually activate transcription. So that's how I think of it. I don't think of it as being like really structured and organized. I think of it as being more of this sort of loose, higher concentration type of thing. But I think it is really interesting to sort of start to try to think at that level and think what is really going on this one piece of DNA. But I think these are really hard questions to answer. But that's how I think about it. I'm sorry if I ran a little over time. I tried to get as many slides out of my talk as possible while still kind of keeping the importance of our story.
No, thank you so much. I did the rest of the team. And so thank you, Jennifer and Joanna, for both your talks. And this will finish the seminar part